Yeah. So good morning, my dear students. Here I welcome back. I welcome you to NEET World. Here we are going to discuss biomolecules. Okay. So this is from class 12th IPE chemistry part. Okay. Right. So in this session I will be discussing two chapters. One is biomolecules and other one is polymers. Okay. So from this chapter we can expect 2 marks questions. Okay. From this chapter we can expect 2 marks question. Okay. So first of all, let's complete this biomolecules, then we'll move for the polymers. Okay, right. Look at this. The first question here says about what is a zwitterion. Give an example. Okay, so we need to define zwitterion and giving one example. Okay, so yeah, whenever they are asking about zwitterion, you just remember amino acids. Okay, why I will tell you, just listen this. See, an aqueous solution, in an aqueous solution, what happens? The amino acid, see, amino acid, it means for that compound there will be acid group and one more is amine group, carboxylic acid and amine, right now. So one is acidic group, amine group is a basic group. So one acid and base groups are there now. So now what happens is, this acid releases proton, that proton is accepted by this base like NH2 group. Therefore the compound formed after transferring that ion, we can call it as zwitter ion. Okay, let me give you the example here by writing it. Yeah, so let us say there is an amino acid here. Let us say R CH COOH is there. So this is a carboxylic acid. So on this alpha carbon, let us say there is one NH2 group. So amine is a basic group and carboxylic acid is an acidic group. So whenever you place this in the aqueous medium, the proton from this carboxylic acid site will releases and that will be taken by this amine group. Okay, now what happens, tell me? R CH, this becomes NH3 plus. Okay, this is NH3 plus and this becomes a COO minus. Okay, so you got a zwitterion like this right now. So this is called a zwitterion. So what is happening in aqueous solution? An amino acid COOH group loses a proton and NH2 groups accept the proton. Okay, accepts this, forming a dipolar ion called, see here we have 2 poles and 2 ions are there here. So it's a dipolar ion. We can call it as a zwitter ion. What is the name? Zwitter ion. Okay, I hope you got what do we mean by zwitter ion, right? Right. The next one here is, what are essential and non-essential amino acids? Give one example for each. Okay, see amino acids are building blocks of proteins, like these amino acids will form peptide bonds that will be resulting you to the proteins right now. So for making proteins we require amino acid right now. So some of the amino acids are essentially we need to take in our diet from external source. Okay, and some of them are not required. Okay, they are already present. Okay, so now see, those which cannot be synthesized in our body, that should be taken from our food, like from the diet. Okay, those are considered to be essential amino acids. Okay, which can be synthesized by our body itself. So they were considered to be non-essential amino acids. Look at here, essential amino acids means that cannot be synthesized by the human body and it must be supplied through the diet. Those are considered to be essential amino acids. Example here, valine, leucine. Okay, so these are the essential amino acids. When it comes to non-essential amino acids, that can be synthesized in the body from other compounds. Okay, so example, glycine, alanine. Okay, that's it. This is the simplest way you can represent your answer for the 2 marks. Okay, about essential and non-essential. Next, define denaturation as related to protein. So what is denaturation? The term related to proteins they are asking. Okay, so just read these ones. So denaturation is a reversible or irreversible change in the structure of a protein. What is this? Denaturation is a reversible or irreversible change in the structure of protein molecule, resulting in the loss of its biological activities and its property. So for example, when you heat a particular compound like a protein molecule, what happens? It will loses its biological activity or whatever the property it will show. Yeah, that property got lost when we are heating it or when we are cooling it. Okay, so this process can be considered to be denaturation. Okay, so denaturation is a reversible or irreversible change protein molecule resulting in the loss of its biological activity or properties are considered to be denaturation. The next, write any two biological functions of nucleic acid. Okay, so here we are asking, so DNA transfers heredity information from one generation to another generation. Yes, so we are calling it as generation, heredity. Now it comes from grandparents to parents, parents to again their children, again to their children, like that. That is what we are considering it as heredity, right? So that is DNA transfers heredity from information from one generation to another generation. This is one of the function. And the second function here, DNA and RNA helps in protein synthesis in the cell. Okay, protein synthesis in the cells. Okay, so these are the two important points you can mention about the biological functions of nucleic acids. Okay, right. The next one, so these are the phosphorylation can be biomolecules first. Okay, if you have time, yes, you can go through the other questions also, but these 4 questions are must to do from biomolecules. Okay, for 2 marks. Okay, and for 4 marks I already explained in one other session. You just go through that, so that both sessions will give you complete idea on biomolecules. Okay, right. The next here we have one more topic here is polymers. Okay, let's see, there are 8 questions almost from polymers in this material. Right, one more thing, you can access this material for free of cost and make your preparation more effective, okay, using this material. Okay, now what to do for accessing this material for free of cost? Just go to Play Store and download MedWorld app. So that is our NEET World's official app. Okay, from that you just log in. First you log into that app using your mobile number, then you just mention your class and group, so that for example if you are an MPC student, you will get maths, physics, chemistry. Okay, so you just mention MPC second year or BPC second year, like that. So you can get your biology materials along with physics, chemistry for BPC. Similarly for MPC, along with maths you will get physics, chemistry. Okay, right. So now look at this, what is of rubber? Okay, so it's a process of heating of raw rubber with sulfur. Okay, with what? Raw rubber with sulfur, so that it improves the elasticity property of rubber. Okay, so it's a process of heating of raw rubber with sulfur at 373 Kelvin to 415 Kelvin to improve the elasticity of rubber. Okay, increase elasticity and elasticity strength and resistance to abrasion. These are the things we can do with the vulcanization of rubber, that is treating or heating of rubber with the sulfur. That's it, you just remember it. So when we heat rubber with the sulfur, what happens? Its elastic strength increases. Just remember that. Okay, sulfur forms, and what is the reason behind this? So now sulfur forms bridges between the polymer chains and gives rubber better mechanical properties. Now what this sulfur will do? It will forms bridges between the polymer layers. Okay, like polymer chains are there, no? Like polymer chains, between two polymer chains we can have a sulfur bridge bond. Okay, so due to that reason the properties of rubber will be elevated. Okay, then next, what is PHBV? How it is used to remember? Okay, this PHBV means poly beta hydroxybutyrate. Okay, and we have beta, like it's a biodegradable copolymer, you can say butyrate copolymer with beta hydroxyvalerate. So is a biodegradable polymer and a copolymer of 3-hydroxybutanoic acid and 3-hydroxypentanoic acid. What is this? So here you have carbonyl linkage here, carbonyl linkage between these two. Okay, so how it will be formed? From 3-hydroxybutanoic acid, what is the monomeric units here? 3-hydroxybutanoic acid and 3-hydroxypentanoic acid. Just remember, it is 3-hydroxy acid, this is also 3-hydroxy, but it is a butanoic acid, here it is a pentanoic acid. And one more thing, if you are using only one type of monomer, you should say it's a normal polymer, but if you are using two different monomeric units, you can call it as a copolymer. That's it. Okay, once again, poly beta hydroxybutyrate. Here this is CO means carbonyl group, that is the linkage between these two, between beta hydroxybutyrate and beta hydroxyvalerate. It's a biodegradable polymer and biodegradable polymer and a copolymer of 3-hydroxybutanoic acid and 3-hydroxypentanoic acid. That's it, remember. Okay, the next here, uses used in medicine for capsules and control drug release. See, we will be seeing the the medicine like capsules. If you observe, there will be a top layer, so that layers will be made up of this PHBV. Okay, it is used in medicine for capsules, okay, and control drug release. Okay, used in special packaging and orthopedic devices. Okay, these are the two examples, are the uses. So what is a Ziegler-Natta catalyst? Okay, so Ziegler-Natta catalyst is nothing but like lithium, like TiCl4 plus trialkyl lithium, trialkyl aluminium trialkyl. Okay, so it means, okay, see here, it is a mixture of triethyl aluminium, that is a trialkyl, or you just remember it as triethyl aluminium. So Al C2H5 tris and titanium tetrachloride. So TiCl4 plus Al CH3 C2H5 tris. Okay, so just remember triethyl aluminium and titanium tetrachloride. So this combination is called Ziegler-Natta catalyst. So it is used for preparation of polymers, especially high density polyethylene is used for preparation of HDPE. You can remember high density polyethylene preparation. In this preparation will be using Ziegler-Natta catalyst. Okay, one of the very useful catalyst for this polymer reaction. Okay, the next here, what is PDI, polydispersity index? So it's a ratio of weight average molecular mass. Okay, something like we have number average molecular mass, weight average molecular mass for polymers. Okay, so it is a ratio of weight average molecular mass to the number average molecular mass. Okay, that is nothing but the weight average molecular mass by number average molecular mass. You just take the ratio of these two things. Okay, so that ratio is called PDI, polydispersity index. Okay, for most of the polymers, PDI lies between 1 to 1.5. This is a range for most of the polymers. Okay, right. The next one here, what is an addition polymer? Give one example. See, addition polymer, by adding two monomeric units we will get this polymer. Okay, a polymer formed by addition of identical monomer molecules having double or triple bonds is called addition polymer. See, for example, ethene is there. So you take n number of monomeric ethene molecules. Now they will add themselves and you are going to get polyethene right now. So you are going to get polyethene. Similarly, Buna S. Okay, so a polymer is formed by addition of identical monomeric molecules having double or triple bonds is called an addition polymer. Okay, the next question is, what are fibers? Give example. Fibers are thread-like solids with high tensile strength. What they are? Fibers are thread-like solids with high tensile strength and modulus. Okay, they are long, thin and flexible. So these are thread-like thread-like solids with high thin and flexible. Okay, they are long with thin and flexible nature. The next, and they have high tensile strength. Okay, examples here, nylon, silk, terylene are the examples. Next, what is the difference between Buna N and Buna S? So Buna N and Buna S. So Buna S, see, Buna S. So it is, both of them are 1,3 butadiene, but here it's a styrene, you can remember. So 1,3 butadiene, 1,3 butadiene. But Buna S na, S here you can remember it as a styrene. Here it means you can remember is acrylonitrile. Okay, what is it? Acrylonitrile. 1,3 butadiene plus acrylonitrile polymer is called Buna N. So where it will be used, it will be oil resistant rubber. Okay, it will be an oil resistant rubber. Okay, So here Buna S 1,3 butadiene plus styrene copolymer is called the synthetic rubber, that is Buna S. Okay, these are the things you can remember. Then next, bakelite. So bakelite is a combination of formaldehyde plus phenol. Okay, the monomers here are formaldehyde and phenol for bakelite. Write the names of monomers of the following polymers. For bakelite, it is formaldehyde plus phenol. For nylon 6,6, it is adipic acid plus hexamethylene diamine. Okay, what is this? Adipic acid and hexamethylene diamine polymer is called here nylon 6,6. Okay, the next, what are polymers? So polymers are macromolecules made up of monomeric units right now. So polymers are large molecules that are macromolecules formed by repeated linkage of small units called monomers through covalent bonds. Example, polythene, polyvinyl chloride, PVC. So where do we use this PVC? PVC pipes are there now? Yeah. So the next, nylon 6,6 and Buna S. Okay. So that's it. These are the 9 questions from polymers. You can expect one in the exam for 2 marks question. Similarly, I have discussed from biomolecules also. In this session it's
B is the final.
Answers are now clear. If you have any confusion, please mention in the chat box. Okay, and remaining topics we will be discussing in the upcoming session. So stay connected with NEET World channel, and if you like the content, please hit on the like button, and you can share with your friends who are preparing for IPE exams. Okay, so thanks for watching. Stay connected.